Okay, let's be quick. Now, in order to be quick, I'll go directly to the question. Like I'm going to read AI, for example, but of course, on the exam, you read the question from the beginning. Fine. So now you have a cell, magnification 2500, and state the evidence from figure 1.1 that shows that the cell is from the stem of a plant and not from the leaf of the plant. Okay. Noor, can you tell me why is this plant cell coming from a stem and not from a leaf?
Yes. Wait, it's coming from a cell, not from a leaf. A
No, my
stem.
dear, it's a stem, not from a leaf. Plant leaves, what's their main function?
Photosynthesis.
Exactly.
As
Now, hmm,
chlorophyll.
okay, do you see chlorophyll or you do not see chlorophyll?
No, I don't see chlorophyll.
Yes, there is no chlorophyll. I don't also see chlorophyll. These are different cell organelles, and the nucleolus is here, and vacuoles. And so it's not a plant cell that does photosynthesis. So I'll write this down. So that's just no chlorophyll. Okay, no chlorophyll. Now, the next table here, we are asked to complete each row in the table to identify a cell structure in figure 1.1 that carries out the function listed. Now, tell me, Adham, which name of cell structure will help in gas exchange from the picture here? Gases, where are they exchanged in a leaf, in a cell like this? And don't tell me stomata and guard cells because I do not see stomata and I do not see guard cells.
Airspace.
Excuse me? Airspace?
Airspace. Yes.
My dear, air spaces are between the cells. You do not see between the cells. You are looking on inside one cell. This is a transmission electron micrograph. So you are looking inside the cell. You're not looking between the cells. Air spaces are between the cells. I'm looking inside the cell. I can see the whole cell with all the structures inside it. Which structure within this cell helps in the exchange of things in general? Gases, liquids, solids. Hmm.
يعني لو letter F.
F, what is F? F is what? F is this. This is a vacuole. What do you mean? The vacuole exchanges gases or the which part of the vacuole or how is F related to
I
the
thought it's an
exchange of
airspace
gases?
برضه بس I thought يعني إن إحنا we need it inside of the cell يعني فكان لازم تبقى F مش airspace.
No, 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 no. Plant cells don't have air spaces inside them, they have vacuoles. There are air spaces between the plant cells. So if this is a spongy mesophyll cell and this is another spongy mesophyll cell, the air space is here. You are looking at the cell inside it with all the organelles. So which organelle here helps in the exchange of materials in general, Adham? Gases,
D.
liquids, solids. Excuse me, my
D.
dear?
D.
D?
Which is a
D?
vesicle.
What is
Yes.
D is a
It's a
vesicle?
vesicle.
Okay. D
D.
is wrong. Because D, if it is a vesicle, I'm not sure what it is actually. If it is a vesicle, then it will transport things from one place to another. Exchange
Okay.
happens across what? Exchange of
Across
things. Yes, yes, you're right. Across.
the membrane.
Yes, you are correct. Across the membranes. Okay. Which letter is the membrane?
A.
Yes, you are correct. It's A. It's an easy and a tricky question at the same time because we all know that cell membranes controls what moves in and out of the cell and the exchange of materials happens across the cell membranes. But because we are looking at a plant cell and they asked you for gas exchange, your brain will go to stomata and guard cells. So the first thing students will look for is a stomata or a guard cell or an airspace. So you're not abnormal, you're normal. They're just tricky. They're just being tricky. So the answer is A, actually, which is the cell membrane. This is, or any other membrane that will let me highlight to this question. Gas exchange. Okay. So the name of the cell structure will be the plasma membrane or the cell surface membrane. Fine. And the letter is A. And we'll go over to the mark scheme and check. Okay. Now, the production of subunits of ribosomes. Where are ribosomes produced? Noor, where are ribosomes produced here? Now, this is easy. Which organelle inside the cell produces ribosomes? Produces ribosomes, not proteins.
النيوكليس.
Not the nucleus, no. Something inside the nucleus.
nucleolus.
Yes, which is which letter?
nucleolus is C.
C. Okay, so this is the nucleolus. Nucleolus, I think this is with a U. And the C. Active transport. Again, this is easy. Which structure here helps in active transport?
A.
Yes, correct. Also A. A is the cell membrane. Any membrane actually contains carrier proteins and helps in active transport. So the cell surface membrane or the plasma membrane. And again, it's A. Aerobic respiration. Now, this is easy. Which here organelle is clearly the mitochondria? Kareem, where is the mitochondria that does aerobic respiration?
At E.
E. Yes, it's E. Okay, so it's the mitochondria and E. Okay, now I just want to look at the mark scheme to make sure that the first one is correct. So this is which exam? 1 minute. It's A. Okay. Cell surface membrane. Good. This so I don't get confused. Fine. Now the next question. Again, I'm not going to read it because it takes so much time, but you will read it. You obviously see here a process of what? What's the process that you see here? Where's Mariam? I cannot hear her voice.
Yes, doctor.
What do you see here at X? They will ask you your name, the process that is occurring at X. What do you see here at X?
Exocytosis. Oh,
Wait, look at the arrow. Yes, it's endocytosis. Okay. This is endocytosis. Okay. Explain how proteins that have not folded correctly are broken down in the vacuum. So they're telling you here definitely that the unwanted proteins are broken down. Now, how these unwanted proteins are broken down in the vacuum? What do you say? This is easy. How are proteins broken down?
They're broken by hydrolysis.
Yes, very good. They're broken down by hydrolytic enzymes. Okay, or hydrolysis, which is the same thing. Now talk about this reaction. The reaction, it needs water or it releases water?
It needs water.
It needs water. Okay. What else will you say? You have 3 marks, so what else will you say?
Enzymes, the enzyme protease will bind with the proteins, forming enzyme substrate complex to break them into amino acids.
Very good. Okay. The protease will break down protein into amino acids. Very good. You can add something else. What are the bonds that will be broken down?
Peptide bonds.
Very good. You have to mention at least 1 or 2 types of bonds in a protein that will be broken down. You have the peptide, you have the hide bonds, the hydrogen disulfide ion, all the bonds will be broken down because the protein contains all the bonds and anything will be broken down. Okay, I think this is enough for 3 marks. We mentioned that this is a hydrolysis reaction. Hydrolysis, you need water, protease enzyme. Of course, the name protease is very important. It will break down the protein into amino acid. It will break down the bonds. Good. This is easy. Describe the role of the lysosomes in animal cells in defense against pathogens. So this is a direct question asking you about the role of the organelle lysosomes. Adham, what is the formation? What is the function of the lysosome? What is the role of the lysosome?
Lysosomes contains hydrolytic enzymes which destroy the pathogen. They contain lysozymes, carbohydrases, nucleases and proteases.
Excellent. So, and you gave examples. Okay. But there is something that you missed. And you gave examples. You said carbohydrates, you said lipases, you said proteases, and say what they break down. Okay. So
Yes,
proteases break down
that's
proteins,
what
lipase break down. Lipase breaks down which bond?
ester bond.
Correct. Now, there are two other enzymes that you usually forget. There's an enzyme called the nuclease.
Nuclease,
Yes, these nucleases, what do they break down?
the nucleus.
No,
Nucleolus.
no, no. The nucleases, they break down the
DNA.
nuclear
bonds
amide.
لا
inside the phosphodiester bond. Nucleases break down phosphodiester bond. Okay. And there is the
lysozymes.
lysozyme. These break down the peptidoglycans in the cell wall in bacterial cell walls. Okay. So if you know those, you're sure of those, you write what you know. But these lysozymes also help in a particular process. There is a name that you missed. We said it contains hydrolytic enzymes and it breaks down blah, 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 blah. Yes, it breaks down what? The bonds in what? What do we want to break down actually? These lysozymes, what do they break down?
The cell wall
The cell wall of what? Okay.
of the pathogen.
Of
The
the
pathogen.
pathogen, exactly. This is what I wanted you to say. So it breaks down or it kills the pathogen. Now, this process of breaking down the pathogen and killing the pathogen, this process has the name. When I kill the pathogen,
Phagocytosis.
exactly. This is what I wanted to hear, the phagocytosis. Actually, this is also an indirect question that is asking you what is happening in phagocytosis. Okay, this lysosome, if you remember, you have the neutrophil. The neutrophil will engulf the pathogen, and then this is phagocytosis. And this pathogen will be put inside a vacuole called the what? Phagosome, if you remember. And then this phagosome will fuse with the lysosome to produce the what? Phago,
Lysosome.
Phagolysosome.
phagolysosome. Okay, I know that you know all this information, but you don't understand the concept of these examiners. They want to trick you, they want to misguide you. So you already know the steps of phagocytosis. I know that you do, but they did not ask you a direct question. What are the steps of phagocytosis? They can easily do that, but they did not do that because they are misguiding you. They want you to be confused and not know exactly what they want. This is the idea. So they did not ask you what are the steps of phagocytosis. No, they asked you the question in a different way. Describe the role of the lysosome. You will mention, like what Adam said, these lysosomes contain hydrolytic enzymes, examples, blah, blah, blah. And these hydrolytic enzymes, they break down unwanted material or pathogens. And then you will go to the mark scheme and you will see that they wanted the term phagocytosis, phagolysosome. Why didn't you ask directly? They don't. Okay, so let me show you. If we go to the mark scheme, here they want the phagolysosomes described, except fusion with phagosomes, phagocytic vacuole or vacuole. So they wanted the steps of what? Phagocytosis. They did not ask, they did not say directly. We said all this, breakdown or digestion of pathogen bacteria, we will kill them. You will name two enzymes, you will name the substrates that are hydrolyzed to form harmless or useful soluble products. So we said that, but it did not cross our mind to write the steps of phagocytosis, even though this is what they wanted. And it's one of the main parts of the answer. 1 and 2 have to be there, and 3, 1 of 3, 4, 5. Do you understand? That's why I keep on telling you 100 times, write everything, okay? Because their questions are not direct, their questions are indirect. That's the problem. So I'm sure Adham knows what is phagocytosis, and I know that he knows the steps. It just did not cross his mind. Okay, Adham?
Yes, doctor.
Okay. Glycogen and cellulose are polymers, and figure 2.1 shows small representative regions of a glycogen molecule and a cellulose molecule. So here you have two molecules. Which one is cellulose, the first one or the second? Oh, it's just written. It's written. It's written there. Okay, describe three ways visible in figure 2.1 in which the molecule of glycogen differs from the molecule of cellulose. Kareem, can you tell me how the glycogen is different from the cellulose? This is the glycogen, and this is the cellulose. What are three visible features? Hmm. Kareem,
Can I say?
where is Kareem? Where is Kareem? I just want to know. Kareem is gone? Okay, Noor, of course you can. What can you see in a glycogen that you don't see in a cellulose?
First, glycogen is
Of
branching.
course. Okay. And the cellulose is? Mm,
It's not branched.
it's
It's not
parallel.
branched. Parallel. Yes.
Okay. Two, what else do you see in a glycogen?
The glycogen is made of alpha glucose
Very good. It's a polymer made up of alpha glucose monomers.
monomers.
And what
Yes.
about the monomers in a cellulose?
Monomer beta glucose.
Yes, correct. It's beta glucose. Beta glucose. Okay, what else do you see in a glycogen that you don't see in a cellulose?
The glycogen has 1,6 and 1,4 glycosidic bonds.
Very good. You see the branches
The
are 1, 2, 6 glycosidic
Yeah,
bonds at the branches.
cellulose it has only 1 to 4.
Yes, very good. Very good. Excellent. Perfect. So this is an easy question. Now, explain how the structure of glycogen is related to its function inside cells. So you know that the glycogen is branched and it looks like this, and this helps it in its function. We took it. What is the function of glycogen and how does this structure help it in its function? Who will answer? Where's Mariam?
Yes.
Okay. Describe the structure of glycogen first.
The glycogen is made of alpha glucose and it is branched.
Yes. And how does this help it in its function?
Glycogen is used in storage, therefore, the branching makes it easy to break down to provide energy for the cell.
Very good breakdown. That's it. Okay, easy to break down, and when it's broken down, it provides energy. And these are the terminal molecules. Remember the molecules that are found at the end. I remember the day when we were explaining this in class. These are the terminal molecules that are easy to break off to release energy and easy also to add on. Easy to add on. And when I add on monomers, I'm going to be releasing energy or storing energy.
Storing.
Storing energy. Okay, it's incomplete, Mariam. So
Can
you
we
said it's
say that
branched. It
it's
helps in storage.
insoluble? Can we say that the
Very
protein is
good.
insoluble
It is insoluble. That's very important. Any storage molecule, starch, glycogen, it has to be insoluble. Why?
so that it doesn't cause changes in the water potential?
Yes, does not no osmotic effect. Great. Does not cause changes in the water potential. Okay. Also, when it is branched or coiled, it is easy to store. You can say it is compact. It takes less space. Okay. I don't, nothing else is crossing my mind. Very good. This is an easy question. Explain how the arrangement of the cellulose molecules in plant cell walls is related to their functions. This is a very important question and it's direct. What is the function of the cellulose and how does the structure of the cellulose help it in its function? Adham, can you please remind me what is the function of the cellulose?
Forms the cell wall of the plant cell
Okay, and then why is it an ideal molecule to form the cell wall of plant cells?
because it has very high tensile strength, uh,
Correct.
which can help it support the stem of the plant while
Hmm.
while withstanding the pressure made by water.
Perfect. Yes, so it will withstand This is the main function of the cell wall, that it does not rupture where when there is high turgid pressure. Okay, this is the function. Okay, when there is high turgid pressure, the cell wall will not rupture. And I just want to stress on tensile. High tensile or mechanical strength means the cell wall stretches or it does not stretch. It is inelastic.
It's elastic.
No, it's not elastic, my dear. Anything that is tensile means it is inelastic. It has high mechanical strength. It doesn't break. Even if it stretches a little bit, it doesn't break. That's why when the plant is filled with water, maybe it will extend outwards a little bit, but it is not going to break because it has high tensile strength. You cannot break it. Okay, so it is inelastic. Inelastic cell walls are inelastic with high tensile strength. It is not easy to break. You can bend it, but you cannot break it, and it will withstand the high turgid pressure inside plants. Okay, this is the function. Now explain how the arrangement of the cellulose molecules make it strong. What will you discuss here?
It has many hydrogen bonds between
Yes,
each
you will
cellulose
discuss
molecule.
actually the structure of the cellulose, everything. It is made up of a polymer, beta glucose units that are, every other beta glucose unit is inverted, and this inversion creates lots of what? Just like you said, Noor,
Hydrogen bonds.
many hydrogen bonds, of course. So you will say that the every other beta glucose unit is rotated or inverted. This is only found in cellulose. Fine. And this increases the strength. Why? Because it will create many hydrogen bonds, just like you said. What else? Many cell, and then they are branched or parallel. Continue the structure.
Parallel.
They are parallel. You have many cellulose molecules that are parallel. 60 or 70 cellulose molecules together, they create a what? Hmm?
Microfiber.
Very good. A microfibril, not fiber, microfibril. And many microfibrils create cellulose
Fibers.
Fibers.
fibers. Very good. And these fibers, are they arranged or they are oriented in an arrangement or in so many different directions?
Many different directions.
Yes, these fibers are arranged in so many different directions, creating the large pores in the cell wall of plant, also making the cell wall of plant freely or semi-permeable.
Semi-permeable.
Cell wall,
Really permeable.
Uh,
really permeable. Okay, so this is a direct question that directly depends on how much you studied. You studied cellulose, you will answer it perfectly. You did not study cellulose, you did not answer it perfectly. Okay, because this is in your syllabus. This is in your studying material. So here you will mention that the cell wall is of high mechanical strength, high tensile strength. It will resist rupture of the plant cell when the plant cell is turgid. Why? Because it has the molecular structure that will help it to do so. And you will simply list everything about the structure of the cellulose. And you can even draw a small diagram. And this question can be out of 3 or out of 6. That same question, because there are so many things to say. The parallel arrangement, polymer, beta glucose, rotated 180 degrees every other beta glucose, many hydrogen bonds creates high strength. It will create microfibrils, and then it will create fibers that run in so many different directions. And there are large spaces between them. That's why the cell wall is freely permeable. So you will write the whole paragraph, that page in biological molecules about cellulose. Okay. Always they get a question about either cellulose or collagen or hemoglobin. Those three things are very important. Cellulose, hemoglobin, and collagen. Those three. And they get direct questions about their structure. So please study them well. We are question 3 now. So state why membrane proteins are required for the movement of molecules such as glucose across the cell surface membrane. Easy question. State. Give me an answer. Why do we need membrane proteins? Mariam, they help in the crossing of what?
The polar molecule.
Yes, polar molecules, charged molecules, like example, glucose, amino acids, because the cell membrane is hydrophilic or hydrophobic.
Hydrophobic.
Hydrophobic, shatur. Okay, now we have a graph here. We have the concentration of glucose on the x-axis and the rate of uptake of glucose. I have to read the question, unfortunately. Researchers investigated one type of hexose transporter. Hexose transporter is definitely a membrane protein known as I don't know what, which is found in the fruit cells of this plant. They used a mutant strain of yeast that has very few hexose transporters in the cell surface membrane to investigate the properties of this protein. The researchers inserted molecules of this protein into the cell surface membranes of the mutant strain of yeast. Equal volumes of mutant cells with this protein were kept in 8 different concentrations of glucose, and the rate of uptake of glucose by the yeast cells in each solution was determined. If I am you, I will underline this. So this is the dependent variable, what they are measuring. They inserted a membrane protein in yeast, and then what did they measure? The rate of glucose uptake by this yeast. All the solutions were kept at the same temperature and pH. So these are the constant factors. So the concentration of glucose is increasing on the x-axis, and as the concentration of glucose is increasing, they were measuring the rate of uptake. Before I move on, tell me, what do you say about this part of the graph? And what do you say about this part of the graph?
In the beginning, the concentration of glucose was the limiting factor. صح?
Correct. Very good. And then what do you say about this part of the graph?
That it's not the limiting factor and it reached
It's not.
Vmax.
Yes, correct. It's not the limiting factor anymore. Something else is the limiting factor. Most probably, if this is the substrate and this is a reaction, then what will be the limiting factor here in B? If, if the
Enzymes.
glucose, yes, the enzyme. Okay, the enzyme, if this is a reaction and the substrate concentration is the limiting factor here it's no longer a limiting factor. I have lots of substrate, lots of substrate, lots of substrate, but I don't have enzyme. The enzyme has been saturated. All the active sites are used up. Okay, explain how the results in figure 3.2 provide evidence to support this conclusion. So I have to read the question. The researchers concluded that this is responsible for the facilitated diffusion of the glucose into the cells. This membrane protein is responsible for the facilitated diffusion. Explain how the results in figure 3.2 provide evidence. Now, this is so clear. When they had this membrane protein, what happened to the rate of glucose uptake?
It increased بعد كده it reaches maximum constant.
It became constant, okay? Because what is going to be the limiting factor at B? It's not an enzyme then. The
ال
researchers concluded
VVH.
that this, yes, okay. So they were adding, so this was present, they added this. I have to read the question again. Researchers investigated the type of the transporter which is found in the fruit. They used a mutant strained very few hexose transporters in the cell membrane. The researchers they inserted this transport protein into the cell surface membrane, and equal volumes of the mutant yeast of this protein were kept at 8 different concentrations. So this protein is like the enzyme, as if it is the enzyme. This this protein was found, it was a constant factor, it was not increasing. It was found in all the yeast cells. They increased the concentration of glucose, and then at one point, the rate of glucose uptake became constant. Why? You understand why now? We have a cell membrane and we have the glucose channel. At the beginning, the glucose was moving inside, and then the amount of glucose moving inside became constant. Why? Because these glucose channels, they became saturated. They are working at their maximum speed. It's like the enzyme became saturated, but it's not the enzyme that we are studying. We're studying the carrier proteins. These carrier proteins cannot admit any more glucose. They are admitting a constant amount of glucose, even if the glucose is increasing, because they are working at their maximum speed. Do you all understand that?
Yes.
Okay. So you would say, why Do the results provide evidence to support this conclusion that this protein helps and facilitated diffusion? Because at the beginning, when the concentration of glucose was increasing, the rate of uptake was increasing. The protein channels were working very well, and the concentration of glucose at the beginning was the limiting factor. And then it became constant, which means that the concentration of glucose is no longer a limiting factor. But what is the limiting factor? The carrier protein. We need more carrier protein so that they admit more glucose. Do you understand, everyone? Mariam,
Yes.
Kareem, Adham.
Yes.
Yes, miss.
Okay. So this is as if it's the enzyme. We will speak about it exactly like if we are speaking about an enzyme, but it's not an enzyme. It's a carrier protein that also helps in the uptake of the glucose. The researchers thought that the grapevines could be modified to have more hexose transporters to increase the size and quality of grapes. So the more this carrier protein is present, the more the size and the quality of the grapes. Explain why increasing the number of these transporters could be commercially important to the growers of grapevines. Again, it's an easy question.
Grapes might be more sweet يعني أو more have
Yes, you
more
are
taste.
right. Of course, the more these transporters are found, the more the uptake of sugar. So I will have the more the uptake of sugar. Now, this has been confirmed. And the more the uptake of sugar, the more the grapevines will taste sweet. They will taste sweet. And of course, when they taste nice, they will sell more, they will be more profitable, and it will bring them more money. Okay, easy question. State what is meant by a non self antigen. Adham, can you tell me what is a non self antigen?
A foreign protein from outside the body.
Yes, very good. Foreign antigen. Okay, it's a protein attached to a carbohydrate. It can be a glycoprotein. Okay, if this was out of 2 marks, what do you say else about non-self antigen? What does it provoke? Does it provoke or it doesn't provoke an immune response?
Which provokes an immune response.
Yes, it does. Okay. With reference to figure 3.3, explain how T lymphocytes respond to infection by a specific type of pathogen. Okay, you don't even need a figure to answer this question. This question is asking you about all the steps of immunity. What happens when your body is encountered with a foreign antigen? From the very beginning, what do you say? This question can be 4 marks or 6 marks. So, what does the T lymphocyte do when there is a foreign antigen?
It has receptors complementary to the antigen.
Yes, you are correct. If it does
Well,
have receptors complementary to the antigen, what will happen?
they bind to the antigen. يعني الانتيجين bind to the T lymphocyte.
Okay.
With this is antigen presenting cell. It becomes an antigen presenting cell. Then it undergoes clonal expansion.
Yeah.
و it releases cytokines. لا
Mhm.
ال T helper cells releases cytokines.
Mhm.
و بس ده اللي انا فاكره.
Okay, no, your answer is incomplete and it is not organized. Okay, let me tell you why. Because you started off by saying that the T lymphocytes will bind to a complementary receptor. Okay, and then you went and you talked about the T helper. Now you talk about the T helper first. Remember the steps. First, you have a foreign antigen. This is the foreign antigen. It will enter the body. Fine. What is the first thing that will attack it? What's the
Is it the
first line
antigen?
of defense? No, no, this is the antigen. X is the antigen. It's the foreign antigen. What's the first thing that will attack the foreign antigen?
الفاجوسايت.
Correct. Which one of them is the antigen presenting cell? The neutrophil or the macrophage?
الماكروفاج.
Correct. So the first thing that will attack the antigen is the phagocyte. Let's remind each other. We have two phagocytes. We have the neutrophils and we have the monocytes or the macrophages. The neutrophil will do complete phagocytosis. The monocytes or the macrophages, they do complete or incomplete phagocytosis.
Incomplete.
Incomplete phagocytosis. And when this happens, they change into an
Antigen presenting cell.
very good antigen presenting cell. So now we have a monocyte or a macrophage with part of the antigen that has remained, and it is an antigen presenting cell. This antigen presenting cell will activate which cell?
ال T
Huh?
lymphocyte
Okay, which type?
helper.
Very good. And the T helper will release,
Cytokines.
correct? And these cytokines will do what?
الcytokines they will
Hmm.
form well ثانية they undergo clonal expansion.
Yes, these cytokines, they will activate the T lymphocytes or the B lymphocytes or more macrophages. In this case, we're talking about T lymphocytes.
Mhm.
Here, the T lymphocytes will become activated.
Yes. هو which
Okay.
one undergoes clonal expansion
The T lymphocyte here.
Okay.
So the clonal selection and clonal expansion actually happens after the phagocytosis. First, we have the entrance of the antigen, and we have inflammation, by the way. We have increase in blood flow, and this is going to bring the phagocytes. Phagocytes will do phagocytosis. And in phagocytosis, we will have antigen presentation. After antigen presentation, this means that the T helper cells will become activated. It will release the cytokines. The cytokines will activate the T lymphocytes. These T lymphocytes, when they are activated, they will undergo clonal selection, clonal expansion, and differentiation. In clonal selection, we will select which T lymphocyte that has a complementary receptor. Here, okay? After we select which T lymphocyte has a complementary receptor, it will multiply by mitosis, and this will be clonal expansion. I'll make more and more and more. And then it will differentiate. And the T lymphocytes, when they differentiate, what do they differentiate into?
T helper و T
Very good.
killer.
Yes. And some of them will remain in the blood and they will
T
become,
lymphocyte.
yeah,
Uh,
some of them will remain in the blood and become what? Memory cells. Memory cells. Okay. So this is actually written in this order in your notes, and I resent you, if you remember, I resent you an organized version in this order exactly. So this is also a direct common question that can be 4 marks or 6 marks or 10 marks because it includes actually half the lesson. What happens when a foreign antigen enters your body? Let's revise it all from the very beginning because this is very, very important. And you will tell me until the final stage. And all of this, all what we said, is the primary or the secondary immune response. Where is Mariam?
Yes, doctor.
All what we said, my dear, happens in the primary or the secondary immune response?
Primary immune response.
Primary. Okay. So tell me what happens from the very beginning. I have an answer. And then this antigen, what does it do? A foreign antigen.
It stimulates an immune response.
Yes, what is the immune response that will be stimulated? In what order? What will happen?
First, the phagocyte is stimulated, or the macrophage.
Very good. And then?
It undergoes phagocytosis, and it produces then an antigen-presenting cell.
It will change into an antigen presenting cell. Very good. And then?
This will stimulate the T helper cells that
Yes.
produces cytotoxic
Cytokine.
cytokines.
Yes.
The cytokines will stimulate more macrophages, B lymphocytes, and T cytotoxic
And T lymphocytes. Yes.
and T
T
lymphocytes.
lymphocytes here, when they are stimulated, what do they go through?
They go through the clonal selection, clonal expansion.
Very good. Colonal expansion and then
differentiation.
differentiation. And you will write something below each one of them. What do you say in colonal selection?
The specific T lymphocyte.
Very good. You will select the specific T lymphocyte that has a complementary huh,
Receptor.
receptor. And after I select, what will happen in colonal expansion?
They will divide by mitosis to
Very
form
good.
more of
After I divide by mitosis, it will differentiate. The T lymphocyte differentiates into what are the different subtypes of T lymphocytes?
T helper, T cytotoxic, and T memory cells.
Very good. The T cytotoxic, what does it do? We know what the T helper does.
They kill the pathogen directly.
Very good. What do they produce to kill the pathogen directly? Perforin or hydrogen peroxide. They make holes in the cell wall of the pathogen or just kill it directly. This is enough. Okay. Now, this is how you write a nice answer, an organized answer. In the mark scheme, it throws keywords in a very unorganized way, but you have to write it in order. When you come to write it, you have to write it in a proper order. Let's look at the mark scheme. I know they're messy. Where are we? This is which number? I think it is 3. Okay. Any 4 from. Except T cells for T lymphocytes, the group or clone of T lymphocytes, they have complementary receptors. We said it. Specific T lymphocytes bind to antigen. We said it. Select T lymphocytes divide many times, undergo colonal expansion. We said it. T helper cells secrete cytokines. Look at this. They told you here colonal expansion and colonal selection and then colonal expansion. And then they said what? T helper cells secrete cytokines. This has to come first. So the T helper cells, they have to release cytokines, and then this will lead to colonal selection and colonal expansion. They are in the mark scheme. I want you to understand that this mark scheme is made for the fellow teachers so that they have a guide while they are correcting. As long as any one of those points are present, they will take a mark. Okay, but this is not the model answer. This is a guide. That's why the mark scheme in any of the sciences looks messy, because it's not model answer. It's just a guide to the fellow teachers that tells them this has to be there, this has to be there, this has to be there. Okay? So detailed effect of cytokine, functions of cytokines, we said it. The T killer cells, they bind they destroy pathogens, we said it. Detail of the action of the T killer cells, we said it. Production of memory cells, we said any one of those, any 4 of those has to be present for the examiner to give you a grade. Fine, but when you write it and while you are organizing your thoughts and remembering the answer, you have to write it in order. Are you with me? So you will not write it in points, separate points like this. You have to write the whole story in order. So at least you remember what you're writing. Okay, Noor? Do you understand the difference between what we said, what you said, and how we answered it?
Yes.
Okay. Explain the meaning of gene. What's a gene? This is so easy. What's a gene? Come on. I'm speaking to myself. What's
Part
a
of
gene?
the DNA, part of the DNA that consists of a sequence of nucleotides that
Of
code
bases
for a
for
polypeptide.
Shatur. Okay, now this is obviously semi-conservative replication. Describe the roles of DNA polymerase and the DNA ligase in the replication of DNA. This is a typical direct question that is not direct. Again, they did not ask you, what are the steps of the DNA replication? They did not ask you a direct question, but they asked you to describe the roles of the DNA polymerase and the DNA ligase in the replication of DNA, which is the same. So you will write in order nicely what are the steps of the DNA replication. Now, this is a very easy direct question. Who will remind me quickly? What are the steps of DNA replication? Tell me, Mariam.
First, the unzipping of and unwinding of the DNA.
Yes, unzipping and unwinding. Very good. And then?
This is by the DNA helicase enzyme.
Okay, very good. Huh? And then?
Then the DNA polymerase enzyme, it joins to the 3' strand of the DNA and
Okay.
starts forming the uh, complementary strand
Okay.
from
How?
the 5' to the 3' end.
Okay. It attaches to the 3 prime, like you said. Okay. How does the DNA polymerase create the complementary strand?
It forms phosphodiester bonds between the
Okay.
complementary nucleotides.
Yes, are made. Huh? And if it is moving, which will be the lagging and which will be the leading strands?
The leading strand is the strand where it is formed directly,
Yes.
and the 3' strand.
Okay, where it will, okay, it was going to move towards the replication formed and it is produced continuously or discontinuously.
Continuously
Good. Now the other strand is produced in,
in fragments,
very good. This is called the,
Okazaki fragments, and it's the
yes,
lagging
the lagging strand, away
replication fork.
from the replication form. And then these Okazaki fragments, who will fuse them together?
The DNA ligase enzyme.
Ligase enzyme. And then what is the last sentence that you write here to end the story of the DNA replication?
This is the semi-conservative DNA replication, where one old strand is present from the original strand and a new strand is formed.
Very good. You gave me an answer that will give you a 5 out of 5, but you just forgot one point. How does the DNA add the new complementary nucleotides? Because they are activated, remember? Free activated DNA nucleotides, they arrive from a nucleotide pool and they are activated because they have two added what? This is the nucleus. They have, yes, two added phosphate groups. And these two phosphate groups are removed by the DNA polymerase. And this generates energy that is needed for the building of the new complementary strands or the sugar phosphate backbone of the new complementary strand. Good. Just don't forget this part. And this activated nucleotide is also called phosphorylated nucleotide because it has two extra phosphates added to it. State the name of the stage of interphase in the cell cycle where DNA replication occurs. Quickly, very easy.
Synthesis phase.
Synthesis, of course. This is an easy exam as long as you studied for it. Okay, Adham. Kareem, where's Kareem? What is structure A, Adham, and what is its function? What is A, my dear? What is A?
Centromere. Centromere
Excellent. What's the function of the centromere?
holds the two sister chromatids together.
Yes, it holds them together. It holds them in the metaphase plate, whatever. What is B?
Spindle fibers,
Yes. And what is the function of the spindle fibers, Kareem?
separating chromatids.
Excellent. Separate them. During what? Which phase are they
Yeah,
separated?
metaphase.
Habibi,
Anaphase.
they are separated in anaphase. Metaphase, they lie in the metaphase plate or at the middle, okay? And they are separated in anaphase. Okay. Complete the figure to show what happens to the chromosome in anaphase. So how do you draw that? You have lots of drawings in your notes, lots of drawings. How do you draw sister chromatids being separated? Hmm. Hmm. Can you draw? Can you draw, or I'm the only one who can draw? Okay, guide me, Noor, guide me. What do I draw? What am I drawing here?
ده which phase؟ ده عضل
Anaphase.
السبندل fibers. Okay.
Yes, so I'm drawing the spindle fibers. Tell me, these spindle fibers, they are attached to what?
centromere.
Very good, they're attached to the centromere.
They
And the
pull
chromatids,
sister chromatids away from each other.
very good. It looks exactly like this. Very easy to draw. They are attached to the centromere, and the sister chromatids are being pulled towards the opposite poles of the cell. Nice. This exam doesn't finish. The last question. State the evidence visible in figure 5.1 that identifies the cells as red blood cells. Okay, very easy.
There's no
Why is
nucleus.
this a red blood cell? No nucleus. What else? Give me another.
No organelles برضه no organelles.
Yes, no organelles, nothing. What else? Look at the shape. It's very obvious. Hmm. Adham, what's the shape of this cell that tells
Biconcave.
you it is a red? Yes, it's biconcave. Now, something
Concave.
else tells you that it is inside a capillary. What if they're asking you, why is this a capillary? Hmm.
Narrow lumen.
Yes, the capillary is very narrow. It's just as wide as the red blood cell. And this is a red blood cell because it is just as wide as a capillary. Look at this. This capillary cannot hold except one cell at a time.
One cell thick.
Yes, the cell wall, the lining of the capillary is one cell thick. Yes. This, if we're talking about the capillary, this is the red blood cell because it is as wide as the lumen of the capillary. So it's very narrow. It's just as wide as the capillary. And what is the width of the red blood cell? Who remembers? In micrometers, not in nanometers.
7.
Yes, yes. Remember this number. 7 micrometers. There's around 7 micrometers. Okay, easy question. Let's finish. Explain how the structure of the capillary is related to its function. Believe me, this is an O-level question. What are the adaptations of a capillary?
One cell thick.
Yes, and why is this important?
To allow
Short distance.
yes,
Mm,
to diffuse short distance and a short
Yes,
distance.
shorter diffusion distance. What else? I want 3. We took this in O-level. This is how we draw a capillary. What are these? My
It's
goodness.
porous.
Yes, it's porous to allow for gas exchange. And the width of the capillary, wide lumen or very narrow lumen?
Very narrow lumen.
Narrow.
Of course, very narrow lumen. Why?
Because it's 2 micrometers thickness.
Number 1, it's 7 micrometers. Number 2, the lumen is very narrow so that the blood flows quickly or slowly.
Slowly.
It will slow down to give time for diffusion. If you remember, this is O-level. Okay. And the lining of the capillary, is it smooth endothelium or what? I'm giving you answers. Hmm? Smooth endothelium. The capillaries are made up of smooth endothelial cells. Why smooth? So what will flow inside the capillary smoothly? I'm giving you
Blood,
answers.
red blood cells.
Yes, smooth endothelial cells or smooth endothelium, so that the blood will flow smoothly inside the capillaries. They're porous to allow for gas exchange, and they are lots of capillaries or little capillaries. Lots of capillaries are always present at the capillary bed to increase the what? My goodness.
Surface area.
Yes, to increase the surface area. Okay, so we have always lots of capillaries to increase the surface area for lots of supply and lots of exchange of materials. And a lot of materials are exchanged. You can give examples: glucose, amino acids, waste products, white blood cells. It helps in phagocytosis because the white blood cells can squeeze their way out of the pores. Okay. Explain how an increase in carbon dioxide in blood leads to the release of more oxygen from red blood cells. Who will answer this question for me? Again, it's a direct question. By the way, this exam is easy. It's not difficult. You studied well, you will answer this exam really well. How increase in carbon dioxide in the blood leads to more release of oxygen from the red blood cells, more dissociation. I'm speaking to myself. Hello, Noor.
Okay.
Uh,
More
by the
height.
way,
More. Yes.
this question here, they are nice and they gave you a diagram to remind you, but they can get you this question without a diagram. So what are they talking about?
First, more carbon dioxide will diffuse into the red blood cell.
Yes, you are correct. It will diffuse out of the muscle and into the red blood cell to be carried. Okay, and then?
And more carbonic acid is formed.
Okay, you jumped. How is it formed? So here is the diffusion part, correct. From the carbon dioxide will come out of the muscle and it will diffuse inside the red blood cell, correct. They gave you a diagram to remind you, even though they can ask you that same question without the diagram. And then the carbon dioxide will do what?
Condensation reaction.
Habibi, what are you saying, Noor? What are you saying? Please
Uh,
don't give me a heartache or an increase in blood pressure. Carbon dioxide reacts with water.
yes, yes.
Okay, this is how carbon dioxide is transported around the body. In the presence of which
Yes.
enzyme? Who will catalyze this
Carbonic
reaction?
anhydrase.
Yes. So they are actually being very generous and they gave you a picture to remind you of this equation. But you can be asked this question without a picture. And when this reaction happens, what is produced?
Carbonic
What?
acid.
Ah, correct. And then it is dissociated into what and what?
Hydrogen and um, uh, hydrogen carbonate.
Yes, yes, yes. Hydrogen ions, as you can see, it is charged,
Yes.
and hydrogen carbonate ions. What is the fate of the hydrogen ions? These hydrogen
They
ions,
react with hemoglobin عشان they have
exactly.
higher
Why?
affinity.
Yes,
Uh,
bravo. If they don't react and they are left in the body, what is their problem?
um,
Why are the hydrogen ions very dangerous? They will make the blood
they
what?
change ال pH بتاع البلد. They
Of
decrease it.
course. Yes. So they gave you a picture actually to help you, to tell you. Here, they just did not show you that the hydrogen ions react with hemoglobin. So that when the hydrogen ions react with hemoglobin, what does it give you to create what?
Hemoglobinic acid.
Exactly. And for the hemoglobin to react with hydrogen, the hemoglobin, it will have to dissociate, leave the oxygen, go out, because this oxygen will be given to the tissues because the tissues are respiring. Take oxygen and give me the hydrogen ions. Okay? So you will simply write the story of how the carbon dioxide is transported. It's a direct question. Nothing is tricky about this question. And you can also talk about the chloride shift. Why does the hydrogen carbonate ions go out? What will move in instead? Give me an
Chloride
answer.
ions.
Yes. And don't give me an answer when you are not sure, Noor. How many times did we say that? The chloride ions will move in to prevent the charges building up inside the red blood cell. So it remains neutral. So this is the direct question. Direct. Carbon dioxide reacts with water in the presence of carbonic anhydrase enzyme, which is present in the cytoplasm of the red blood cell. It will give you carbonic acid. It will dissociate. We said this 101 times. Explain why the concentration of the chloride in the plasma of the deoxygenated blood is lower than in the plasma of oxygenated blood. What happens to the chloride ions? Why does the concentration of the chloride ions drop? Tell me, Mariam, where does the chloride ions go? We just answered this question.
Yes, doctor.
Mariam,
Yes.
where does the chloride ions go?
When the hydrogen carbonate ions leave the red blood cell,
Yes. Yes.
the chloride shift occurs and the chloride ions enter
Yes. Very good. Why?
to keep a stable charge.
Very good. So that the red blood cell does not become positively charged. Good. Okay. Now, this question, again, plants. They have to get a question about plants. The structure labeled X is part of the symplastic pathway. State the name of structure X. What is X? X. This is very easy. What is X? Kareem. What is X? Adham.
Plasmodesmatic.
Shatur. Plasmodesmata. Okay. The structure Y is what? What is Y?
A peristan strip.
Yes, the Casparian strip and the suberin deposition. Good. Now, with reference to figure 6.1, complete the statements. Water moves from the soil solution to the cytoplasm of the root hair
Osmosis.
cell by osmosis. Yes, this is easy. Water moves from the exilum in the root to the leaf. How does the water move the exilum?
Mass flow.
Excellent. By the mass flow. Okay. Water moves from the mesophyll cell walls to the intercellular spaces. Here, the water here, it will move from the surface of the mesophyll cells into the intercellular spaces.
Evaporation.
Evaporation. By the way, here also, water moves up the exilum. We can say several things. We can say mass flow or something that is very common, transpiration pull. Don't forget the transpiration pull, okay? Due to cohesion and adhesion and changes in hydrostatic pressure. Yes, evaporation. Here, evaporation. Water vapor moves from the intercellular A spaces to the outside by? How does the water vapor go out of the stomata?
Diffusion.
Diffusion. Okay. This, by the way, is a question that I got in O level in one of their quizzes. So this is an O level question, actually. Oh, it's finished. We finished this exam. I was actually thinking that they will ask us about the movement of the transpiration up the water up the exilum. There's so many things to say, and the symplastic pathway and the apoplastic pathway, but the question ended here. So this exam, in my opinion, it's a very easy exam, but it depends on your direct studying. You studied well, they asked you direct questions. Let's look exactly what they asked you about. There was a lot of molecular biology here. They asked you about the cellulose and the full structure of cellulose. Please, before paper 2, study cellulose, the structure very well, collagen, because you are usually confused between cellulose and collagen, and hemoglobin. In every paper 2 exam, they have to ask you an essay question about one of those three: cellulose, collagen, and hemoglobin. The structure and the function and mutation in terms of hemoglobin, everything. And also they asked you about phagocytosis, the steps. They wanted all the steps of phagocytosis. Direct question: immunity and molecular biological molecules. Again, the what's this called? The glycogen. This was a graph question, easy. They did not get any question on enzymes. Cell membrane, they did ask you about cell membrane a little bit. And DNA replication, this was a direct question about the semi-conservative replication, a little bit of mitosis, and here a little bit of transport in mammals. The direct question of how do you transport carbon dioxide, which is half of the lesson anyway, and the chloride shift, and a very small question about plants. So this is an easy exam. You studied well, you will do well. And if I want you to notice something, again, all of you give me incomplete answers, not because you did not study well, because you do not know the question, you don't understand the question. All the questions are tricky, they're not straightforward. So when they ask you about something, write everything about the related topic, everything. Like when it happened with Noor, the lysosomes. He told me what the lysosome does when they wanted the steps of phagocytosis. Did you understand that? Are you with me? Any questions?
No, thank you.
No, thanks.
Okay. I still feel that you need to study harder. Please study harder and solve as much as you can paper 2. Focus on paper 2 now. I'll meet you on Wednesday at the same time. Bye. Send me if you have any questions. Let me see. Where am I?